Okay, so first for today, I would like to acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, language, and cultures of First Nation, Métis, and Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. We would like to thank our sponsors, and this includes our speakers today, who will also support us for our research day, which is happening on March 26th. As a reminder, please all, if you haven't so, register for the 2025 ATI Research Day. But we're welcoming today Zeba Malik and Shana Labers from the company OLink, who are their Western Canadian representatives and application scientists and specialists for the area this company is working in, which is an academic spin-off company, as I learned from Scandinavia, and was recently acquired by Thermo Fisher Scientific based on a very innovative technology. And therefore, we felt it was of interest to the research community here at the U of A to have Zeba and Shana come to us and share with us insights of this novel technology, which is, from my perspective, truly different and innovative. So Zeba is the business development manager with OLink for Western Canada for the Midplex products. She is a trained molecular biologist with a PhD in molecular life sciences from the University of Zurich in Switzerland, and spent 2 years as a postdoc here with the University of Alberta, and is located also here in Edmonton. So if you, after this presentation, want to reach out to Zeba for a meeting and learn more about the technology, that is easily to facilitate. And then switch to commercial businesses, and 5 years ago, been with OLink for since then, and now OLink is, as I mentioned, transitioned into the Thermo Fisher group of companies. Shana is a senior field application scientist for OLink Proteomics. She provides the scientific support to customers, including project design and data review, as well as supports laboratories with the implementation and training of the OLink workflow in their facilities. Shana has more than 15 years of research experience in the field of proteomics, genomics, biobanking, and also clinical research. And today they will focus on the research community. And I know OLink also presented to our clinical chemistry lab partners, because this technology has probably applications in both areas, research and clinical diagnostics. And the title is, as you can see, Accelerating Biomarker Research with a Unique PEA Technology. So with that, I'm handing it over first to you, Zeba, and yeah, take it away, please.
That's right. Thank you. Thanks, Michael. Thanks for the introduction, and thanks for giving us the opportunity to present. A little correction there. So we did come out of a lab from Sweden. So our headquarters are in Uppsala, Sweden. And last year we got acquired by Thermo Fisher Scientific. Shana has control over slides. So Shana, I'll say next, and then if you can forward the slides for me, thanks. So next slide. So first things first, declaration of conflict of interest. We are both associated with OLink Proteomics. As Michael mentioned, I am business development manager for our Midplex portfolio for Western Canada, and Shana is our FAS for Canada. Next slide. The objectives very quickly. The goal of the presentation is to give you a real-world example, of how OLink's PEA technology is used in some of the research areas. To this audience, I think the interest is more in transplantation and some, immunotherapy. So we'll cover those topics first. We have selected some case studies. The idea is to give you a high-level overview of how PEA is used in these studies, and then go on and discuss what our technology is and some of the technical aspects of our technology. So this is the agenda. Our presentation is divided into two parts. Essentially, the first part is case studies, as I mentioned. Shana and I both are going to go back and forth on this. I will be presenting a transplant-related case study, and Shana will present immunotherapy. And then I'll take over, and then I'll present PEA. So we'll start with the immunotherapy ones. Shana, please take it away.
Hi, hello everyone. So I have two case studies here to present with you today. The first one here is an example of how our OLink Target 96 assay can identify a protein signature that can be used for patient stratification and disease prediction. So this study here by Nunez and colleagues uses a multi-center cohort collection collected both in Switzerland and Germany to evaluate the development of immune signatures that could predict the onset of autoimmune toxicity in patients that are treated with immune checkpoint inhibitors, or ICIs. Here, they used a combination of mass cytometry and OLink to look for markers associated with serious immune-related adverse events, or IRAEs, in both melanoma and non-small cell lung cancer patients. So briefly, this group used the Target 96 inflammation panel, which consists of 92 unique markers on this panel, to evaluate serum samples from both non-small cell lung cancer and melanoma patients who were treated with ICI therapy. From here, they divided it into three subgroups. So those that did not respond to the ICI treatment and did not acquire ICI toxicity, those that did respond to ICI treatment and did not experience ICI toxicity. And then lastly, those that did respond to treatment but experienced toxicity as well. And these were serum samples that were collected over 4 time points. So pre-therapy administration, 1 to 2 weeks after ICI therapy, and then at the onset of autoimmune toxicity. From here, the Target 96 panel was used to analyze a total of 172 samples, 28 from non-small cell lung cancer and 24 from melanoma patients. So as you can see, the results of the OLink analysis are highlighted here in these violin plots from both melanoma on the left-hand side and non-small cell lung cancer on the right. Here you can see the fold change between 2 time points, time point 1 and 2, that are on the x-axis, or sorry, the y-axis there, and each patient group, which is on the x-axis. So if we look at the third group, we can see those that exhibited a response to the ICI treatment and also experienced immune toxicity for both cancer types. And we can see the highest fold change in expression is between these 2 time points. So here in this analysis, they found 4 proteins of significance with this pattern: CXCL9, 10, 11, and interferon gamma. You can see by these yellow boxes that these trends were similar in both cancer types and were significantly higher relative, or significantly higher, had a significantly higher relative increase observed in patients with those in melanoma. What's interesting here is the early increase in interferon gamma related proteins in the serum was only observed in responders with immune toxicity and not in the non-immune toxicity patients. This suggests that the protein signature can act as a predictor for those who develop these immune-related adverse events. Additionally, they were able to validate these findings with a multi-center cohort of 362 serum samples from melanoma patients. And here the same protein signature predicted the development of ICI therapy induced immune-related adverse events, independent of whether they were responders or non-responders to the therapy. So the outcome of this study expands the understanding of the pathomechanisms of immune-related adverse events and provides first evidence in identifying candidate biomarkers that can predict the risk of ICI therapy responders developing these immune-related events. Of note, since the study also used cytometry to identify some key T cell subsets, that were also likely to be associated with an increased risk of these immune-related events. They were able to combine the results from both to create a powerful risk assessment tool for the early identification of patients who had higher risks of seeing these adverse events occur. And this is important because even though it will not prevent the development of these adverse events, it is often associated, as it's often associated with tumor responsiveness. But these markers can act as predictors. And so we can help support and improve patient care through more effective monitoring or any new needed interventions. This next example is investigating cytokine release syndrome, or CRS, an immune effector cell-associated neurotoxicity syndrome, or ICANS, with patients that are receiving CAR T therapy. These therapies have revolutionized the treatment options for B-cell malignancies. However, common adverse events occur, such as CRS and ICANS. And despite their frequency of occurrence, the biology of both conditions is not completely understood. Currently, the best predictor of these events is disease burden. So prior to the CAR T infusion, this means that the current best predictor of these events is looking at disease burden prior to receiving the CAR T infusion. This would mean that higher disease burden would equate to a higher risk of, higher risk of severe ICANS or CRS. But this metric currently has poor specificity. So this group at Philadelphia's Children's Hospital and the University of Pennsylvania were looking to leverage proteomics in efforts to reduce the suffering in patients that had CRS and ICANS. Additionally, there has been some early indications in prospective clinical trials about early intervention using cytokine blockades to help mitigate the risk of severe CRS in these patients. So therefore, this group was looking to identify novel or potential biomarkers that could either predict the severity or act as targets for possible therapeutics. During this study, this group used OLink Explore panels to measure 1,463 proteins from serum samples collected at multiple time points in both pediatric and young adult B-cell acute lymphoblastic leukemia patients. This was looked at prior to and following CAR T infusion in two groups. The first here is the 26 patients, 13 with minimal and 13 with severe CRS. The second subset of patients was analyzed 732 proteins using one of our OLink Explore 384 oncology panels and inflammation panels. And this was using an expansion cohort of 17 patients. So the results of the proteomic analysis identified two markers of interest: FMS-like tyrosine kinase 3, or FLT3, and mast cell immunoglobulin-like receptor 1, MILR1, as both perfusion predictive biomarkers of severe CRS. During their analysis, they found that patients who developed severe CRS had initially high levels of MILR1 and FLT3, and then these levels would decline over time. Going one step further and and this was done using linear regression to help identify these proteins. And during their analysis, they actually noted, based on the ROC curve here you can see on the left, that the predictability using these markers to better understand disease, the severity of CRS, outperformed the clinical standard, which was using that disease burden calculation. So these results were also corroborated in their analysis using RNA sequencing and flow cytometry. Additionally, they investigated the pathophysiology of CRS and ICANS, and during this analysis, they evaluated differentially expressed proteins and looked at the detectability of cytokines within these pathways. Here, they noted associations of CRS with the interferon gamma pathway. And interestingly, 2 other novel biomarkers were identified as potentially targetable cytokines that were associated with ICANS. And these were IL-18 and CELA3A, as you can see from the plots on the right. So we can see here with this study that the results from the proteomic analysis can identify biomarkers that outperform clinical standard tests. In this case, they identified and validated 2 proteins that could serve as candidates for pre-infusion serum biomarkers to predict the development of CRS. Interestingly, the one protein FLT3 inhibitors have been extensively studied in pediatric populations, and this data suggests that it could then be further investigated for evaluating the predictive value for CRS and highlights the potential use of drug repurposing. Even more, the MILR1 marker has not been previously reported in pediatric leukemia biology. So therefore, in this analysis, they identified a novel marker for the detection and new potential treatment options. Lastly, the other notes in this publication, which I thought was interesting, is that the serum biomarkers that they identified could actually obviate the need for the current practice of using pre-infusion bone marrow aspirates for CRS prognosis. And so they indicate that serum markers may be better at defining disease burden than using just a single aspirate, as the disease burden could be dependent on the site that was taken. So this is an interesting point, I think, as well, because having the ability to identify blood biomarkers using non or less invasive methods such as serum as opposed to aspirates could really go along with patient experience and patient care. Additionally, the findings of the study show that identifying potential targets for further therapeutic development and drug purposing opportunities. So we saw here with the IL-18 using targetable cytokine in other inflammatory conditions. We could use IL-18 here as well for blocking therapies, and therefore could be easily translated to clinical trials involving ICANS. Lastly, using blood biomarkers patients can be assessed prior to infusion to better understand the severity of CRS and ICANS and offer better treatment options to reduce patient suffering. And with that, I'm going to pass it back to Zeba.
Thank you, Shana. So I decided to go for this publication as this is one of the most recent ones that showcases PEA in transplantation. This was published just a couple of months ago in December of 2024. This was authored by Dr. Jason Goldberg from Inova Shar Heart and Vascular Medical Center in Virginia. The title of the publication is Proteomics in Acute Heart Transplant Rejection on behalf of the graft investigators. So the main theme of this research publication was proteomic analysis and identification of corresponding molecular pathways that are associated with acute cellular rejection and antibody-mediated rejection. Next slide, please. So I wanted to touch upon what GRAFT is. GRAFT is the Genomic Research Alliance for Transplantation Study. This study was published a few years ago, back in 2021, and is very well documented and has been cited over 200 times. And the figure on the left shows the study design. I added this just because it's using that study design and it's using patients from this study. So I think it's important to see what they did and what GRAFT is really. So GRAFT is a prospective multi-center longitudinal study that enrolled heart transplant recipients before transplant and followed them serially after transplant, including patients aged 18 years or older. They excluded patients undergoing repeat or multi-organ transplantation. This study compared the donor derived cell-free DNA of acute rejection to controls to determine test characteristics. This current study that I'll be talking a little bit more about is focused on proteomics. And the idea was to identify signatures between acute cellular rejection and antibody-mediated rejection. 104 heart transplant patients were included in this analysis from the GRAFT study. Patients with and without rejection had a similar distribution of age, sex, race, and BMI, as well as pre-transplant diagnosis. All demographic and clinical characteristics are listed in this table on the right. This study included 18 patients with ACR and 12 with AMR. 4 of the AMR patients had a history of prior ACR and 3 presented with mixed rejection, both antibody-mediated and acute cellular rejection as their first episode of rejection. Next slide, please. So what was the challenge? Why did they decided to do this? So there's increasing use of molecular methodologies that are currently being used to detect cardiac allograft rejection. 40% of adult recipients are hospitalized for this acute rejection within the first 5-year post-transplant, and nearly 50% will develop cardiac allograft vasculopathy, which is a manifestation of chronic rejection by 10-year transplant. Contemporary post-transplant biomarker surveillance include donor-derived cell-free DNA, which has a high negative predictive value and mild to moderate positive predictive value for both acute cellular rejection and antibody-mediated rejection. Additional biomarkers include gene expression profiling, which is limited to ACR only, and recently microRNAs, which both identify ACR and AMR. However, there is currently very little information on the in vivo inflammatory and cardiovascular injury protein profiling associated with rejection. So the opportunity here was to perform a blood-based protein expression profiling that will allow for simultaneous evaluation of hundreds of proteins. This will evaluate immune responses, inflammation, myocardial injury, and vascular disease. Prior studies have shown an association between protein expression profiling and other transplant outcomes, specifically primary graft dysfunction and cardiac allograft vasculopathy. Identification of differentially expressed proteins during acute cellular rejection and antibody-mediated rejection can aid in understanding the biologic mediators and protein pathways for each rejection type, as well as the pathophysiology of rejection. So moving on to what they did and what they found. Next slide, please. So they looked at 181 proteins using 2 Target 96 panels from OLink. These proteins were evaluated from the blood drawn at the time of endomyocardial biopsy. Protein fold change, logistics regression, and pathway analysis were conducted. So differential expression was found in 2 proteins during ACR and 73 proteins during AMR, as shown in the volcano plot in the middle. In ACR versus no rejection, there were 2 proteins, the inflammatory proteins, CXCL10 and CD5, with significant expression in ACR. In AMR versus no rejection, there were 73 proteins with significant expression in AMR. In ACR versus AMR, there were 3 proteins, NT-proBNP, ST2, IGF-BP2, and CX3CL1, with significant expression in AMR as compared with ACR. And there was 1 protein that was found with significant expression in ACR as compared with AMR. Next, differential expression between AMR and ACR is shown in the box plots on the right. And NT-proBNP, ST2, and IGF-BP2 remained significant predictors for differentiating between the rejection subtypes. Additionally, CX3CL1, which was upregulated in AMR, and PON3, which was upregulated in ACR, have both been implicated in atherosclerosis and heart failure. There have been also multiple descriptions of CX3CL1 in lung and kidney transplant rejection previously. Also, expression of both CX3CL1 and PON3 have been identified in patients with cardiac allograft vasculopathy. ACR has been understood as a T-cell mediated myocardial injury that, attracting C helper cells, natural killer cells, and monocytes, resulting in phagocytosis and apoptosis. This aligns with this current study's identification of cell-mediated immunity markers, which is CXCL10 and CD5, and pathways of T-cell receptor signaling and immune cell differentiation. So this data of CXCL10 and CD5 has been presented before in renal transplant rejection, but heart transplant rejection evaluations were limited to animal models. Also, an interesting finding was IGF-BP2, as there have been limited evaluations of this and other insulin-like growth factors in heart transplantation, but these biomarkers have been associated with graft rejection and dysfunction in renal transplantation. Moving on. So here, the ROC plots show proteins that had the 10 highest fold change for acute cellular rejection and antibody-mediated rejection as compared to no rejection. So the authors mentioned that even though they were not looking for biomarker analyses, they just wanted to look at protein expression profiling. ROC analysis did show an AUC of 0.73 in evaluation of CXCL10 as a marker of ACR, as well as AUC of 0.80 in the evaluation of NT-proBNP, ST2, CPA1, CPB1, and IGF-BP2 as markers of antibody-mediated rejection. The evaluation of these proteins as biomarkers of rejection, of course, it requires further study in appropriately sized cohorts, specifically the association between NT-proBNP and AMR, which have a significant association in this study, but were not associated with AMR in prior studies. The figure at the bottom shows the gene set enrichment analysis for both ACR and AMR. This, together with pathway analyses, indicated significantly different pathophysiologies for the two types of rejection, with cardiac injury markers predominant for antibody-mediated rejection and pro-inflammatory markers for acute cellular rejection. Pathway analysis identified T-cell receptor signaling and cell differentiation as key pathways in ACR and cardiovascular disease, and cell turnover in antibody-mediated rejection. Next slide, please. So the solution was that this was a novel proof of concept evaluation of proteomic data in heart transplant rejection that showcases differential protein expression during both AMR and ACR. And why does this matter? Of course, it's the authors of this publication, they suggest that donor-derived cell-free DNA does give quite a lot of information, but additional insights into the pathophysiology of rejection, especially AMR, if you want to look into that, it's important to look into protein expression profiling. And lastly, to conclude, protein analysis reveals unique biomarkers and biological pathway expression in both ACR and AMR. Okay, that was the case study on transplantation. I want to move on to the next slide and share a little bit about this project, the Pharma Proteomics project. I'll be spending maybe a couple of minutes here, but I think it's important. It's a large scale proteomic initiative by UK Biobank. This study is called Pharma Proteomics Project, and it's the world's largest proteomic study. Our PEA technology was chosen as a preferred method out of the existing techniques to perform proteomics on 54,000 samples from UK Biobank cohort of 600,000 individuals. Next slide, please. So to give you a bit of an overview on timelines, the UK Biobank was established in 2006. This generated one of the world's most extensive databases from around 600,000 participants. In 2020, UK Biobank Plasma Proteomics Project was established using OLink's PEA technology. This was to plasma profile 3,000 proteins in over 54,000 participants within this database. This is a longitudinal study, so this database is also enriched for 119 diseases. There's data from genomics, MRI scans, a lot of different data. And now there is also proteomics data on the first 54,000 samples from this cohort. So just in 3 years, towards the end of 2023, the results of this proteogenomic initiative was published in form of 3 Nature publications. UK Biobank, Biogen, AstraZeneca, and partners, and Decode Genetics authored these revolutionary Nature publications. Next slide, please. So all of these, next click. So all of these publications share a common experimental design. They leverage the genomic data from UK Biobank and proteomics from UK Biobank Plasma Proteomics Project. So I'm not going to go in detail, but if anybody's would like to go and dive a little bit deeper into these, I'm happy to share articles and chat a little bit more about this this initiative. Next slide, please. So, and I'm happy to share that UK Biobank will be moving forward performing proteomics on all of the 600,000 participants using OLink. This is very important, as this data will be available for researchers to data mine and look into, and so this will be really amazing for the research community. Moving on. So this was the case studies. Now I want to talk about what our assay is and who we are, and sort of, a little bit about our company. Next slide, please. So as you can see, our headquarters are in Uppsala, Sweden. Our North American headquarters are in Boston, but essentially we are spread across North America, Europe, and Asia. And we have become an established market leader in proteomics field. We have a strong validation with more than 2,400 peer-reviewed publications. And we are also involved in clinical trials around the world. Next click, please. Yeah, so that's the image on the top right. So this number is a little bit outdated. We are involved in around, I think, 100 clinical trials at the moment, and the circle or the pie graph on the right shows the breakdown of different disease types that we are involved in. Next slide, please. So in this image, I'm showcasing our scope. So our scope is uniquely, all the little dots in that, the blue box are the little proteins that are part of our library. And our scope is uniquely positioned to interrogate the complete dynamic plasma proteome. We have designed our library to cover all protein pathways from classical plasma proteins to tissue leakage markers and low abundant inflammatory proteins with an unparalleled specificity and sensitivity. So if you look at the top box here that says classical plasma proteins, those are the most abundant ones and easy to find, but really the challenge is trying to capture these low abundant proteins. So our technology really is able to capture these low abundant proteins. And as I'll show you in the next slides, it's pretty remarkable that how we are able to achieve that by just using one as little as 1 microliter of sample. So next slide, please. So here I'm showing central dogma of biology, DNA, RNA, proteins. Someone might argue, like, why do I have to study protein? I can just do genomics and, or transcriptomics. But the reason why it's important to look at proteins, it's because, proteins represent real-time biology. They are dynamic and actionable targets in both health and disease. And in central dogma, proteins are closest to us in terms of outcome. It's true, DNA does help characterize our risk for disease from birth, and RNA is a proxy for proteins. But when you really want to look at real-time biology, we need to look at proteins. And it's important to note that over 95% of clinical trial targets are protein. Most of the drugs and OTC medications target protein. So moving on. So why did OLink, come up with PEA? The reason was that they were, obviously with traditional proteomics, there were some challenges like low sensitivity, lack of specificity, high sample consumption, and limited dynamic range. So we wanted to work on these problems, or OLink wanted to work on these problems, and then we came up with PEA assay. Next slide, please. So this is essentially the assay. So our PEA technology consists of matched pairs of antibodies that carry unique DNA tags. Upon binding to target protein, the DNA tags are brought to close proximity. They'll hybridize and later extend to form DNA barcode. When it's but in case of non-specific binding, the oligos will not hybridize and therefore not result in any readout signal. The extended oligos gives a unique barcode for each protein. This extended DNA barcode is ready for amplification and detection using either qPCR for our target products or NGS for our explore products. Next slide, please. Yeah, so this shows the amplification by qPCR on the top in the top image there, or NGS in the bottom image. So again, for a qPCR, the protein expression is directly related to the number of qPCR cycles for our target products. For our NGS readout, the barcode will identify each sample and protein, and there's a digital signal which will translate to protein concentration in each sample. Data processing will result in individual protein profile for each sample or patient that will provide a view of real-time biology. Next slide, please. So we have addressed all of these traditional proteomics challenges using our PEA technology. I don't want to go into it because I think I've already explained it in the previous slide. Next slide, please. So this is a very powerful image, as it clearly shows, the issues of, or the problem with the traditional proteomics. So with affinity-based proteomics, you would get a signal no matter what type of binding event is going to happen, right? With our unique PEA technology, you're going to get a signal only you require matching dual antibody recognition with these unique oligo tags that need to be present, and both the antibodies need to bind in order to create this signal. So this again, the reason when we are able to eliminate the biological crosstalk, that's only when we are able to scale to thousands of proteins without any loss of fidelity, which essentially is what PEA does. Next slide, please. We could see why the market has adapted PEA so quickly, and we are seeing increasing number of publications day after day in high impact journals, and currently we are sitting at more than 2,400 publications in all of these different disease types. Next slide, please. And I think one of the other features of PEA is that you can use the same technology, go from discovery all the way to your unique custom panel of proteins for your disease of interest or your area of expertise, and use the same technology, go up or high in plex using PEA. A lot of times, people are looking just for one golden biomarker, that's gonna, answer all the questions. But it's, in a more realistic term, or in a more realistic setting, it's rather a signature of proteins that's, causing a problem or, issue, or, it's more like a signature that you need to identify in order to answer your questions. Okay, next slide, please. Yeah, one of the other example is that we support researchers, clinicians, other customers in different types of processes. I think I already mentioned this, but we also support different types of clinical trials, and also post-approval surveillance in case you want to go to an LDT route or an IVD route. Next slide, please. Yeah, so I think I didn't stress this enough, but one of the things, or one of the unique features of PEA is that you are able to detect a lot of these proteins, thousands and hundreds of proteins, by using just minute amount of samples as low as 1 microliter. So it allows you to perform different types of omics techniques. Anyone in preclinical space working on mouse, you're able to do longitudinal studies using 1 microliter of plasma or serum from mouse. You can see from this slide that we also have the flexibility of input samples. It's not just EDTA, plasma, or serum. There are a lot of different matrices type where where we have experience in. Yeah, if you have a specific matrix type that you want to talk about, or are there questions, Shana and I will be happy to discuss that. Next slide, please. Our assays go through rigorous quality control. Our validation data is made freely available. You can go on our website and see exactly how we validate each individual assay, and as a product, and the product as a whole. So there are essentially these 5 criterias listed here. We validate our products based on specificity, detectability, scalability, sensitivity, and precision. Next slide, please. Here are some of the things that I've listed, or some of the research areas that our customers are using our product for. So it's understanding biology, improving detection, and develop better therapies. Next slide, please. Reduce risk in drug development, saving time and money. Next one. And essentially precision medicine. So essentially we have a couple of more slides. I wanted to discuss, I wanted to discuss a little bit about OLink Insight platform, which is an open access platform. Yeah, Shana, do you want to take this and maybe speak on it for a few minutes before we end the presentation?
Sure. Yeah. So OLink Insight is a free web-based tool. You can sign up for a profile just using your email address and have access to a variety of different web-based resources. And it's really designed to support researchers end to end within their research studies. So we actually have a series of resources for pre-studies. So looking at disease risk associations, we leverage data obtained from the first pass-through of the UK Biobank at proteomic analysis to evaluate more than 3,000 proteins and assess disease risk associations. We have data stories on normal ranges, looking at 3 different cohorts from 3 different continents and developing normal ranges in proteomics. And then looking at proteogenomic association. So how can we combine data from proteomics and genomics data together to really corroborate that we are identifying or targeting the protein we intend to. So these are all different stories and resources that are readily available at this site. And then from the post-study aspect, we can really look at, which of these disease associations did I locate in my research, as well as, are also corroborated in the UK Biobank data set. We can look at the normal ranges and identify, which features of these proteins might help me explain my results. Do I have elevated or upregulated proteins where in normal or control populations it might not be the case? And then how can we use cis-pQTLs to really create that gene disease association? And so we have data from the UK Biobank to kind of help guide researchers on how they could look at this data as well. And this is just highlighting, that disease risk association tool that I was explaining. The data is there available on the site that you can download and investigate yourself. It's using more than 50,000 unique individuals drawn from the UK Biobank. And these individuals, have both clinical and biological sample collections, like 10-year follow-up. So this is all accessible free for you at any point, as well as a wealth of other resources there as well to help you plan a study. And so with that, I'll end here. And if there's any questions, I think we have a few minutes.
Yeah, no, thank you for that, right? While people are considering questions, what I think it's important, as you said, in the immune and transplant context is that, of course, there is a larger body of literature suggesting that in body fluids, like for kidney and urine and for heart in blood, our chemokines and various proteins are elevated during rejection, and the literature is growing, and it is around for a long time. And what I liked when I first heard about your technology and why I referred you to our clinical lab is that you can custom make panels which are in a higher volume setting, can be used for larger studies like you did with the UK Biobank, right? This is not a cumbersome ELISA plate assay approach. And of course, your advantages you described here about your specificity. But I see there is a hand up by Nafishe. Please go ahead with your question.
Oh, yeah. Thank you so much. Just, as a person that working with cancer immunology, I have 2 questions just related to my own project. The first one is that, that we have always have kind of the problem about the surface protein expression by proteomics and, mass spectrophotometry because, beside the sensitivity, the low expression, the other modification. So I'm just wondering that how your this OLink proteomics kind of the overcome the expression of, detection or measurement of surface protein. And another question is that, most of these research study that you have been shown is using fluid samples like serums, but how about if we want to go through a tumor-infiltrated lymphocyte? I'm just talking about a kind of the cells, not looking about the different expression in the fluid. Yeah, these are my questions.
Yeah, Zeba, did you want to start or?
Yeah, I can start. Yeah, so Nafise, thanks for that question. So to your first question on surface proteins, so the way our library is designed is the references the human, proteome atlas, and we have definitely a bunch of different types of proteins in there, including surface proteins, but we can take this offline and I can share with you, the full list and, discuss which proteins might be of interest and, really go and pinpoint. And the function that Shana mentioned towards the end of the presentation, OLink Insight, there is a very, it's an amazing tool, because where you can put in your list of proteins and it'll pop, it'll give you suggestions on which panel would be suitable for your protein of interest. And, the panels that we have designed are based on specific disease area or, covering a specific pathway. So the idea is to not just look at that one protein or, a set of 5 proteins that you're looking for, but essentially to get ideas and, find connections from a certain pathway or a disease type. Saying that, there's also, an option to customize panels. So we do have a product called Flex where you can combine, a set of different proteins, to run proteomics on that. I think I answered that question, but I don't know if Shana, did I miss anything or you want to add something else?
No, I think you answered it well. I was just going to touch on the sample type that was mentioned. So yes, and some of the examples we did provide today were, liquid biopsy samples like plasma and serum. But we do support a variety of different sample types, and the assay is amenable to different varieties. So anything from like tissue lysates or frozen fresh tissue, cell lysates, cultured media, urine. We've pretty much evaluated most sample types at this point, or worked in collaboration with researchers to evaluate. So, it'd be really interesting to hear what type of samples you have. Also, our scientific support team has a wider range of sample preparation recommendations. So if you did have questions about, is this sample type suitable, please reach out. We can have that discussion and kind of go through the different preparation and see if there's anything that, to see how we can support that.
Okay, sure. And about designing a panel based on the mice protein. So again, you can do that, right? Because all the data that you showed, it was related to human samples.
Yes.
Okay.
So we have already a pre-designed mouse panel. So we have a target 96 and a target 48 pre-designed mouse cytokine panel that is available for use. And so, definitely talk to Zeba after. She has all that list of proteins that could really see if you have the targets of interest.
Okay, thank you so very much. Thanks.
Thanks, John. John put the contact information in the chat for everybody. If you want to follow up with Zeba or Shana or both, you can copy those out of the chat. And also just to add to this, I think, Zeba, you mentioned there is a platform in Alberta or on campus we're already installed.
So it's at UBC right now, not in Alberta yet. But yeah, it would be nice to have one soon. But no, we have it. I have one at UBC currently. So if anybody would like to try running their samples, we definitely have a place in Western Canada to run that.
Yeah, that's also often helpful to just do a trial and see how it suits the own research needs or discovery and follow up biomarker study. Both I think are possible, right? Large scale discovery studies and targeted panel based, population based, or cohort based studies, as we saw in your examples. Okay, so thank you very much again, right, for sharing your insights and spending the time with us. And I hope to see many of you in our upcoming research day on March 26th. And with that, I think we can close today's rounds. Thank you. Have a wonderful rest of the week. Take care.
Thanks so much. Bye, everyone.